Hey everyone, and welcome to the last video for Chapter 4. In this video, we're going to talk about vesicular transport. So this is another active transport mechanism, and in this mechanism, we're using plasma membrane vesicle to transport materials that are too large to move across the membrane. There are two major types: exocytosis, which is a form of vesicular transport where molecules are leaving the cell. And when they leave the cell, they leave within a vesicle that then can just release them out. So think of exiting the cell, okay? And then endocytosis, they're entering the cell. So molecules enter the cell in a plasma membrane vesicle. So we're moving molecules into or out of the cell, and the way we do this is the cell membrane itself produces a vesicle. So either in either mechanism, the plasma membrane is going to be imperative in producing that vesicle. So with exocytosis, remember this is exiting the cell, materials move to the cell membrane, and then this vesicle that is carrying some type of material fuses with the cell membrane and becomes part of the plasma membrane. So I'll show you that in a second, but I'll show you kind of what it looks like. So you have this membrane here, and you have a vesicle. I'm just going to change the color a little, like this, that binds to that membrane. And what happens is you're going to end up having the membrane open up. And so if this is the same plasma membrane, we add on this new color right here from this membrane. So it becomes part of that plasma membrane, and the materials, let's just put some green materials in it, right? We have these little green blobby things are the molecules. Those green blobby things are released outside of the cell. And that's exocytosis. So let's look at what it looks like. And I used green blobby things without even looking at the picture, and there are green blobby things here too. So you have this plasma membrane here, and then you have this vesicle that is moving up to the plasma membrane. The vesicle is composed of phospholipid bilayer, so it just opens up and becomes part of the plasma membrane. Those materials are released out of the cell. That's exocytosis. So endocytosis is then the opposite. With endocytosis, the plasma membrane actually engulfs a portion of the material on the outside. So in this case, I'm just using yellow as my plasma or green as my plasma membrane. And outside of the plasma membrane, we have some different molecules, right? So we're going to have these little blobby things outside, and the plasma membrane is going to engulf them. Let me just get that color back. So let me do this. So the plasma membrane actually surrounds those blobs and pulls them into the cell. That's endocytosis. So the materials enter the cell. And so there are three types of endocytosis: phagocytosis, which we typically think of as cellular eating. So in this case, we're taking in large particles. And not all cells can phagocytose material. So only specialized cells are able to undergo phagocytosis. In the human, we have certain white blood cells that do this. You know what? I'm not going to do it that way. I'm going to put WBC for white blood cells that are able to take in large molecules, specifically macrophages. So macrophages come into the cell or are in your blood, and they encounter, say, a bacterial cell. What that macrophage will do is it will engulf the bacterial cell and pull it in. Then we have pinocytosis, and so this is often thought of as cellular drinking. That is really a K, but it looks really bad, so I'm going to write it again, okay? There we go, drinking. So in this case, we're talking about small droplets of liquid are pulled in, and dissolved in those droplets are tiny solutes. And so this can occur by most cells. So again, in our body, pretty much all of our cells, almost all of our cells can undergo pinocytosis, but very few can undergo phagocytosis. And then there's a specialized form of endocytosis. This is actually a specialized form of pinocytosis. And so I'm going to write that here. And it's known as receptor-mediated endocytosis. And in this case, there are, on the plasma membrane, there are protein receptors. And these protein receptors bind specific molecules. What we always talk about is the cholesterol. So I'm going to put that here. And then those receptors, when they all have bound cholesterol, this entire structure, which forms a, or which then will engulf the proteins and the cholesterol, pinocytizes the material and pulls it in. So pulls in cholesterol on receptors. And so let's look at what that looks like, okay? So here we have phagocytosis, again, a large plasma membrane surrounds a large structure and pulls it in. That's cellular eating. So over here we're seeing single-celled organism like a paramecium being eaten by a larger structure known as an amoeba. This is another protozoan, but it's a larger protozoan that's taking in that paramecium. Pinocytosis, pretty much all cells can do, and so we have this dissolved solutes in a fluid, and the plasma membrane just engulfs that fluid and then pulls in and blebs off. So this vesicle is composed of the plasma membrane. And then in receptor-mediated endocytosis, we have this specialized coated vesicle region that's on the plasma membrane. So this is typically a clathrin-coated region. We have these protein receptors that bind some molecule. What we typically talk about is cholesterol. When all of those protein receptors are bound with the cholesterol, then the entire clathrin-coated vesicle blebs off into the cell. And so that's how we get rid of cholesterol. So that is the end of our lecture. So I'm going to stop here and I will upload these and pull them through Edpuzzle. Have a great day. Bye.